Bismillahirrahmanirrahim. My name is Dr. Shahzad Irfan. I'm presenting my abstract, the fifth International Conference on Applied Zoology 2022, organized by the Applied Zoological Society of Pakistan in collaboration with Khwaja Farid University of Engineering and Information Technology, Rahimyar Khan, Pakistan. The title of my presentation is the temporal assessment of hepatic expression of carbohydrate response element binding protein in alloxon-induced hyperglycemic adult rats. So the first section is the introduction. Carbohydrate response element binding protein, or CRAB, is a transcription factor and is activated by the carbohydrate metabolites. And after its activation, it results in the upregulation of different glycolytic and lipogenic genes in the liver. CRAB is expressed at high levels in the hepatocytes, in the beta cells, adipose, white and brown adipose tissue, and the muscles. And lower expression levels have been observed in the physiologically high glucose levels induce CRAB acetylation. This acetylation leads to an increase in the binding affinity, chemical stability, and transcriptional activity of the CRAB. Glucose 6-phosphate, or G6P, is the primary carbohydrate metabolite, which directly binds to different conserved sites on the CRAB. And these conserved sites are comprised of low glucose inhibitory domain, or the LID, and glucose response activation conserved element, or the GRACE. And both of these domains are collectively known as the glucose sensing module of the CRAB, which implies that the glucose as an important regulator of the CRAB. Now, if we look at the isoforms, the CRAB has two different isoforms, alpha and beta. The alpha is the classical or the canonical isoform, whereas the CRAB beta is expressed by an alternative promoter. The main difference between the alpha and beta domains is that the beta domain lacks the low inhibitory glucose domain, which means that at the low glucose level, the transcriptional activity of the CRAB beta is not affected. Increased carbohydrate metabolism generally activates CRAB alpha and beta, where after the activation, the CRAB beta binds to its own promoter as a transcription factor, and it results in the production of even more beta, CRAB beta isoform. CRAB beta isoform serves as a marker of the CRAB activity because of acute increase in the CRAB beta after the CRAB alpha activation. So this seems to be a very good marker. Increased hepatic CRAB expression, which means both the expression of alpha and beta isoforms, have been reported in diabetes, type 1 and type 2 diabetes, obesity, and metabolic syndrome in humans, which indicates its potential as a diagnostic marker of metabolic status or metabolic syndrome. The current study was designed to assess the CRAB expression of both of its isoforms in a hyperglycemic environment. This hyperglycemic environment in these rats were created by pre-treating the rats within alloxon is a chemical which induces significant beta cell damage, and this beta cell damage do not permit or do not let the pancreas to produce enough of the insulin. So the plasma or the blood level of these animals, a condition known as hyperglycemia. So the material methods, the next section is the material methods. 21 adult male rats were selected for this specific study. These animals were treated with a single intraperitoneal dose of alloxon at the dose rate of 120 milligram per kg body weight. The glucose levels in these animals were monitored through tail vein by glucometer. Because this study was the temporal assessment, which means that we are more interested in the timestamp of the expression pattern of crab. So these animals were sacrificed on multiple days. So these animals were sacrificed on the 0, 1, 3, 5, 8, 12, and 15th day. 0 day is the day on which the Alexon treatment was given, and the 1, 3, 5, 8, 12, 15 day are the post-Alexon treatment days. A total number of 3 animals were sacrificed on each of these days. After the sacrifice, the liver tissue was dissected and collected. Half of the tissue was placed in 4% formalin for histological assessment, whereas the rest of the tissue was placed in a chilled RNA letter solution for the RT-PCR expression. And this expression reaches its maximum level after the 5th day, the expression pattern to the 8th day onward. On the other hand, the expression pattern of the beta isoforms gives us a little bit of a different picture. The initial phase, the up till the 5th day, the pattern is kind of the same as the alpha. But after the 5th day, the significant decline, which we observed in the alpha isoform, is not there in the beta isoform. So this beta isoform, the transcriptional activity of this beta isoform does not seems to decline even in the presence of low glucose levels on the 12th or the 15th day, which means that this isoforms might be responsible for the glycolytic and the lipogenic even at the low glucose level. The other aspect of this hyperglycemia is that the high glucose level eventually induces reactive oxygen species, commonly known as ROS. And these reactive oxygen species, they eventually causes mitochondrial stress and endoplasmic reticulum stress. The mitochondrial stress and the ER stress eventually activates the antioxidant response elements in the genome. The major antioxidant response element genes are the NRF2 and NFKB. In order to observe the expression pattern in response to the high glucose level, we observe the expression pattern of the NRF2 and NFKB. Surprisingly, the expression pattern of NRF2 was quite different from the expression pattern of NFKB. So what we expected was that on the 3rd and 5th day, the expression levels will be high, which was the case as in the NFKB expression. But on the 8th day and 12th day, the expression pattern of these genes were still high. Glucose levels were low. So let's discuss these results. Hyperglycemia was achieved within 24 hours of the single Alexon dose and lasted for 8 consecutive days. From 8th day onward, a downward trend in the blood glucose was observed, reaching almost normal glycemia. Expression profile of the CREB isoforms affirms that high glucose concentration induce upregulation of expression. Hyperglycemia ultimately induced reactive oxygen species with further results in the mitochondrial and endoplasmic reticulum stress. So the mitochondrial and ER stress will eventually activate the antioxidant response elements. This is indicated by the sudden increased expression of NRF2 and NFKB. The timestamp or the temporal pattern of expression reveals that the CREB expression represents a swift response towards hyperglycemia as compared to antioxidant response genes like NRF2 and NFKB. The subsequent decline in the CREB isoform expression on the 12th and the 15th day might be attributed to the decline in the glucose level observed in these days, but that decrease was only observed for the alpha isoform. Activation of NRF2 and NFKB on the 5th day onwards can only be explained by the fact that Substantial cytoplasmic concentration of ROS, reactive oxygen species, because of hyperglycemia was not achieved on the first or third day of Alaxon treatment, which means that the high level of glucose needs some time to eventually produce the reactive oxygen species. And these reactive oxygen species need a little bit of time to eventually induce the mitochondrial or endoplasmic reticulum stress. So this time period during the third day and the fifth day, where we don't see an increase in the NRF2 and NFKB expression, might be due to the fact that during this time, the mitochondrial or the endoplasmic reticulum stress was not induced. So, but again, further data is always necessary to completely understand this complex mechanism of antioxidant response elements and the carbohydrate response element binding protein. Thank you very much. If you have any question, I hope I will be able to answer your question during the actual live online session. Thank you very much.